Hello friends, this video on biomolecules part 16 is brought to you by examfear.com. No more fear from exam. Quick comparison of all the four structures. So when we talk about primary structure, it is just the sequence of amino acid. That's it. Talking about the secondary structure, the sequence gets coiled in this fashion to form helical structure, or it forms a pleated structure like this. Tertiary, this gets further folded. Quaternary, two such polypeptide chains are there, like this red one and this green one, and then they are joined by hydrogen bond. So that forms the quaternary structure. Now let us look at a three-dimensional view of the globular protein. So this is how the globular protein looks like. So if you see, they are highly coiled, and this is the tertiary structure. I mentioned before also, when we talk about globular protein structure, mostly they are tertiary structure because one chain of polypeptides fully coiled and interacting with each other, forming a structure like this. Similarly, if you talk about a globular protein, for example, hemoglobin. So this is how the structure of hemoglobin would be. So this is which structure is this? Primary, secondary, tertiary, or quaternary? This is a quaternary structure because you have two separate chains of polypeptide. One is the blue chain and one is the yellow chain. Two separate chains, they are joined by hydrogen bonds. So this is the quaternary structure. This is your tertiary structure because it forms a spherical shape by too much of coiling. So this is your tertiary structure. Now if you look at this yellow colored structure inside the tertiary structure, that is nothing but your secondary structure. So in secondary structure, what happens? The polypeptides, they interact with one another to form a coiled structure. And inside the secondary structure, if you see the sequence of amino acids, that will look somewhat like this. So this sequence of amino acid is nothing but a primary structure. So this is how the structure of hemoglobin looks like. So now we will talk about another important property of proteins, that is denaturation. Now that we have spoken about all the different structures of protein, you should know that proteins always have a unique three-dimensional structure. So these proteins as such are known as native proteins, but there is a possibility that the protein might lose its complex structure. How? When it is subjected to a change in temperature or pH. Now if you expose the protein, or if you subject the protein to a change in temperature or pH, the bonds, the hydrogen bonds or the intermolecular bonds which we were talking about, they might get disrupted and the globules, helix, everything get uncoiled. So protein lose all its biological activity. The tertiary, the secondary, tertiary, quaternary, all the structures are lost. Only the primary structure is retained. So primary structure, it is just the sequence of amino acids. So the protein is not functional with its primary structure. So protein loses its biological activity and it gets uncoiled. So this is how it takes place. So what happens? Only primary structure is retained. All other structures are lost. And when does it happen? When it is subjected to change in temperature or pH. So then what happens? Secondary, tertiary, quaternary structures are lost. So when these structures are lost, only primary structure is retained and the protein's biological activity is lost. So we can say biological activity is lost. So we will take one example where we can actually see proteins getting denatured. I told you that eggs have proteins, right? Now when you take a egg which is not boiled, a raw egg, what does it have? It has an outer white covering and inside you have some liquid stuff. That's it. But what happens when you boil that egg? When you boil that egg, you get something like this. You get a white portion outside and that yellow liquid which was there inside this forms the yolk. But from where this white portion came? Because outside you just had one covering, but that covering is still there after you boil the egg. You just remove that covering and then inside you have this white colored structure. So this happens due to the coagulation of egg white on boiling. That was because the protein which was present inside the egg, that is the albumin protein. Inside the egg, the protein which was present was albumin. So this albumin protein gets denatured when boiled because it is subjected to a high temperature and a high pH. So what happens? The protein loses its complex structures and only the primary structure is left. And what is that? Once that primary structure is left, it loses all its biological activity and it forms this egg white. And that is why it is said that it is very rich in protein, because its protein is still there, but it is just that it loses all the complex structure and it forms this. So this is an example of denaturation of protein. So with this, I think we have covered our discussion on proteins, and please remember that the structure of proteins are extremely important. Primary, secondary, tertiary and quaternary structure, they are very, very important. So with this, we have covered two biomolecules, carbohydrates and proteins. Now, before we go ahead with the next biomolecule, that is nucleic acids, before that, I would like to have some discussion on enzymes, because enzymes are also proteins, but their behavior is quite different. So we want to study the structure, the function, how enzymes do their job. So we'll spend some time understanding enzymes. Thank you. Please visit www.examfear.com to watch more videos, attempt free online test, get free study material, find tutors and mentors. Thank you once again.